A huge thank you to the Academy of Medical Sciences for asking me to speak today. What an honour that is. And when I looked at the participants on the call today, realised I'm amongst giants. So this is a topic close to my heart, something that I've been involved with for a long time that encompasses a lot of the areas of my interest. Looking at what you might think of as the translational pipeline, but I'm going to propose as the translational circle. What does antenatal care look like in 2021? What's the point of it? Well, broadly speaking, the point of it is to get from a blastocyst to a baby, but needless to say, bumps in the road, and not just for the UK, but this is a map of global maternal mortality, which shows you the health disparities and which is matched by the stillbirth disparities. So what I'm going to be talking about is pre-eclampsia, and whilst the UK has had an important role in the research, for me it's essential that we think about the big picture and about where the global burden of disease is for this condition and many others in pregnancy. This is a paper by Victor Bonney, who was sort of one of the grandfathers of obstetrics, who describes antenatal care in 1914 as measuring the blood pressure and boiling the urine for albumin. And those of you who have done clinical will think that probably not much has changed in all that time, and that's what I still do in clinic myself on a weekly basis. And in fact, blood pressure measurement continues to be a cornerstone, not just in the UK, but around the world, and that's where blood pressure machines exist. And some of our global work is looking at getting blood pressure machines, even the basics, into antenatal care. But we have been busy, and particularly with my colleagues who do more discovery science, looking at how we understand the pathophysiology of preeclampsia, going from the sort of impaired placentation in the first half of pregnancy through into the multi-organ manifestations of the preeclampsia syndrome, with the concept of the soluble FLT and placental growth factor and the angiogenic anti-angiogenic balance being pivotal in this pathway. So this describes the story of what started with my PhD work, where we showed that in normal pregnancy, you should see this typical inverted U-shaped curve of placental growth factor in the second half of pregnancy. Then those women who are destined to get preeclampsia, you see a flat placental growth factor profile. And this was alongside a trial of an interventional trial. But in those days, placental growth factor was a sort of novel candidate biomarker that had promise. But so did many other biomarkers, and it's a question of what made this one special. We went on to undertake a prospective cohort study in women with suspected preeclampsia to look at its use as a diagnostic adjunct. And we showed that with concealed testing for the outcome of preeclampsia requiring delivery within 14 days, placental growth factor outperformed all our other tests in current use. And if I was there in person at the Academy of Medical Sciences, I would show you a cartridge going into a benchtop machine. This is the sort of machine that is used in A&E, for example, for measuring D-dimers. It's readily accessible. And that links back to how I think this might have global reach, because a test that we develop has got to have more than just a high-income setting applicability. Yet again, this was a study undertaken with one of my fellows because I'm keenly enthusiastic about ensuring that we build research capacity in our specialty. So this was a trial of a diagnostic test, recognizing that the huge majority of diagnostic tests that we undertake have never been through trials. And so it was an interesting experience looking at revealed versus concealed, showing that we halved the time to diagnosis when measured in women with suspected preeclampsia, and that we also significantly reduced one of our secondary outcomes, that of maternal severe morbidity. So this was important because it wasn't just that the test was good, we also showed that the clinicians were somehow acting on the results, and it was making an impact on downstream clinical outcomes. How did we do this? Well, we integrated this diagnostic marker into NICE guidelines. We did it on a traffic light system to stratify surveillance. But in an ideal world, we will go back to using this continuous measure because, as with many aspects of biology, it's a continuous measure, not categorized, and look at how we might integrate it further into an algorithm. So what's the impact been? Well, we've had nice guidance, both diagnostic guidance and management guidance for women with pregnancy hypertension to recommend its use as a rule-out test. And that's been assisted by a cost-effectiveness analysis from the trial, which showed a cost saving of £149 per patient, which really comes from women who have a normal result being returned to usual care. Inevitably, in maternity care, we have quite a big safety net, and but actually those women with a normal result can safely return to normal care, with their time to delivery being many weeks subsequently. We got quite a lot of press publicity around the trial results, and that's definitely made an impact in terms of reaching women. And it's also been adopted both by the Accelerated Access Collaborative and then subsequently by the MedTech funding mandate. It's been interesting because when we publish a research paper, we may not always be aware of what happens next for implementation. But for example, watching the pathway through into these NHS funding mechanisms in England has been highly informative for me to understand how it impacts on clinical practice. So for example, the enablement through is one of 4 products that's gone through on the MedTech funding mandate for this year, has been, I'm sure, very helpful for implementation. It's also had particular relevance in the COVID-19 pandemic. This screenshot is taken from a 10-minute video produced by Katie Kurt, one of my other fellows, for the BMJ Awards last year. And one of the things we looked at was how might it be used in the COVID-19 pandemic. So what we really noticed is that in sites that had implemented, they were using it to stratify hospital attendance and footfall. So those with a normal result of a PLGF greater than 100 picograms per mil could return to usual care and reduce the need for repeat face-to-face visits in the hospital, whilst at the same time keeping care at an appropriate level at a time when women were really reluctant to come to hospital, so that we did not see an increase in particularly perinatal deaths in those who are much higher risk. And alongside this, we are also doing the PARAT-2 trial, which is a study of repeat testing. Just because one test is good doesn't mean that blowing the budget on repeat testing is a good idea. So that was ongoing at the moment, and we'll report towards the end of next year. But also woven across this is the concept of health equity. It's been particularly challenging to get pregnant women included in research, both within the pregnancy field and particularly for drugs in pregnancy, and also in relation to other fields. So this was an editorial that we wrote about the concept of health equity. The one exception to this has been the recovery trial, which pregnant women have been actively included in. And that's made a direct impact on the guidance that is being produced by the Royal College of Obstetricians and Gynecologists, which is disseminated internationally. So pregnant women now have access to the same treatments, steroids, tocilizumab, Regeneron monoclonal antibodies, that were shown to be effective because they were included. But both across country settings and just the basic inclusion of pregnant women in research is so important. And there is much more work to do on the inclusion of pregnant women in drug trials, safely, ethically, robustly. Why does this matter? Because we also have disparities by ethnicity. Black and Asian women are several times more likely to die in pregnancy and similarly for their babies. And from work that I'm doing in Ndola with Professor Andrew Shannon and colleagues, you can see that the setting, this is the obstetric high dependency unit in one of the Ndola hospitals, a far cry from what we've had at Guy's and St Thomas's, particularly as we've seen in the last year and a half. So we need to think about solutions that are equally applicable. So maybe we need to rethink. 2 years ago, when I was talking about lateral flow tests, I got a lot of blank looks. Now I think everyone knows what a lateral flow test is, but we need to think about how we adapt our PLGF-based testing to provide it at point of care, for example, across our Zambia settings where we work. So I'm just finishing by talking about the pipeline, because although it's often been conceptualized as a pipeline with the typical valleys where many biomarkers have fallen through into those, I guess for me, it's become more of a circle. And really, I built on the work of many others who had discovered placental growth factor as an integral part of the pathway. And I've had the privilege of with the group, taking it through both the early cohort studies through into a trial into subsequent trials as it gets implemented. But really then asking, where do we start again? So where does the blood test not work? So I'm doing some work with the team at Mercy Perinatal in Melbourne in Australia to look at other biomarkers, both for this group, but also for late-term preeclampsia, where this biomarker works much less well. So as soon as we've reached implementation, it's time to start again and say, what more do we need to do? This is work I'm here presenting, but very much on behalf of the team. And it's one of my joys to build a group and to build research capacity across the disciplines. And particularly, we've had such strong patient and public involvement throughout this research strand. So this is one of our PPI groups. In Lambeth, we have a very diverse multi-ethnic group. And without all of these people, I couldn't have done any of this research. So thank you very much, and I really look forward to taking questions.